Okay, so just before we get started, to remind you that this meetup is sponsored or co-sponsored by NISS and Merck. NISS is a nonprofit national research institute. Among its other jobs is to promote statistical research, including postdocs and conferences and independent research. Merck gets involved with NISS through the affiliate committee. And just to remind you of a couple of items that are coming up on the NISS schedule. One is the 12th annual conference on clinical trials. That should be particularly interesting for this group because they're talking about electronic health records this year, and that's April 17th at the University of Pennsylvania. And then another very important one, many of you know that the American statistician recently had a special issue all dedicated to p-values and significance testing. And to sort of follow on to that, we've gotten a number of experts, including some of the folks who wrote some of those articles. And there's going to be a webinar on May 23rd from 12 to 2 p.m. Eastern Time. You can find information on these two events as well as other events sponsored by NISS at the NISS website, which, as you can see at the bottom, is www.niss.org. And again, I remind you that this meetup is part of a series of meetups that we have basically quarterly. Each of them is hosted, and we have this sort of webinar, but then we post the slides and post the recording at the NISS website. So if you want to catch up on any of the ones that you missed, go to the NISS website and you can find all that information there. Today, I think we have a really distinguished set of speakers, so we're very lucky. This meetup is really a follow-on to the meetup we had last, October, which was on real-world data as well. We got a lot of response from that meetup. Folks who saying they're very interested in the topic, but they not sure how to get started and like to see some basic examples of what people are actually doing out there. So that was sort of the idea behind this meetup. And we really have, as I said, 3 distinguished speakers. We have Mark Levinson from the FDA, we have Dave Madigan from the University of Columbia, and we have Liz Stewart from Johns Hopkins. And so with that, I'd like to pass the ball to Mark and bring up his slides and give him a proper introduction. So Mark Levinson is currently the director of the Division of Biometrics in the Office of Biostatistics, which is part of the Office of Translational Sciences at the Center for Drug Evaluation and Research of the FDA. He's a member of the CEDAR Working Group on developing a framework for the regulatory use of real-world evidence. Dr. Levinson received his PhD in statistics from the University of Chicago and a BA from Cornell University in mathematics. And with that, it's all yours.
Okay, well, thank you, Dan. And I look forward to this session as well, particularly the other speakers. So I'm going to present on real-world evidence and data at the US FDA. We'll present about half of this talk will be on some of the regulatory background. I know it's not quite an example, but I think it might be interesting. And then actually present an example in the second half. So let me see about advancing these slides. Okay, here's a disclaimer because I'm going to be talking about some legal stuff that I'm probably not completely competent enough to talk about, and it will also allow me probably to speak more freely in any sort of questions that come up. Okay, starting, I'd like to present some definitions that I think will be helpful as I go through my talk, and these are actually now somewhat official FDA definitions. There's a distinction between real-world data and real-world evidence. This distinction will sometimes be important. So real-world data is data relating to the patient health status and or healthcare that's collected from a variety of sources. And here are some examples of sources, some of the primary sources.
So Mark, let me jump in because
Sure.
the slides are not advancing on my screen.
What slide are you up to?
We're still on slide 1, and I'm getting notes from other folks the same thing.
Okay. So. Well, I'm on the WebEx, and
okay.
now it's backed up on.
Yeah, I advanced the slide myself, so maybe just for this, if you can just tell me to advance the slides, I'll do it.
Sure.
And we'll see how that works. So I'm up to the FDA definitions.
Okay, so you're on slide 3 now. That's what I'm seeing on my screen now too. Okay, so there was a disclaimer on the previous slide, but it's nothing too interesting. Okay, so now we're on slide 3 about these FDA definitions. So there's a distinction between real-world data and real-world evidence. So kind of the primary sources of real-world data that we're thinking about are electronic healthcare records and claims and billing data, data from product and disease registries, and I'll talk a little bit more about those in the next slide or so. But there are, of course, other sources of real-world data, such as like mobile devices and wearables. I think we're all aware of these. And then on the other side is real-world evidence. This is like using the real-world data to provide evidence of like usage, benefits, and risks associated with medical products. So the data can be used in a variety of designs, including randomized trials or pragmatic trials or observational studies, but it's the final evidence that we get from the use of the data in some sort of design. And naturally, there may be cases where the evidence is derived both from real-world data and more traditional sources of data. Okay, could you advance the slide? Okay, so this is a kind of an illustrative example of a single individual and how various data sources may come into play. So starting off on the left, there's this blue box where this patient goes in for an office visit, gets a diagnosis of hypertension, and receives some dispensing of an antihypertensive. Sometime later, the patient has a particular perspective that they're having difficulty breathing or notice like an increase in heart rate. Further on, the individual goes back, has actually an emergency situation where they have to have an appendectomy and has an inpatient stay and is in the hospital where some other information is collected. And then goes in for another encounter. This time they get a diagnosis of anxiety, and that's sort of concurrent with some feelings that there's some joint pain and some emotional symptoms. Another encounter, the patient comes in and now is diagnosed with influenza and gets some antibiotics, and some clinical information is recorded. And finally, the patient decides to do some lifestyle changes. So some observations on this is, first of all, this patient's kind of having a hard time during this period. But other than that, you can see that there's a number of data sources that come into play. There are these claims data that basically record billing and administrative information. There's electronic healthcare records that kind of contain the more detailed medical information. And even that data may not be in a single source because you both have hospital medical records as well as clinical medical records. And then there's some data that falls outside of these sources. So this real-world data is kind of very disjoint, and no single source may provide all the information. Next slide, please. Okay, so what are the goals of RWD and RWE? That's real-world data and real-world evidence. And I'm mainly speaking for FDA here. Other parties may have different goals. But first, a step back, what traditional randomized controlled trials typically are like. Well, they typically occur outside standard medical practice and procedures. They're like at special centers, or the protocols specify special care or observations of the patients. And they also typically involve select groups of patients. First of all, the patients have to volunteer for these trials. They have to consent. But in addition to that, often trials are done with patients that might be healthier than the typical patient the drug might be intended for. And this is so the drug has a better chance of showing its effect, that you're creating a more sensitive study. And these trials typically involve special infrastructure and data collection. So a whole bunch of like special electronic forms are created to collect this data. So what does RWE try to do? Well, one goal might be to reflect the diversity of patients in actual healthcare practices in the real world. Another goal might be to improve these studies by making use of existing data and infrastructure, like the data sources we saw in the last slides. But with both these goals, FDA is trying to maintain the current evidentiary standards, which I'll get to in a moment. Next slide, please. Okay, I just want to say a little bit about this kind of give and take. I mentioned several goals on the last slide, but they're actually somewhat conflicting. For example, if you have real patients and real health care, that creates a heterogeneous situation. You're going to have more noise, particularly if you throw in some real data there. So all this noise and heterogeneity are likely going to require larger studies to show the same effect. But on the other side of the seesaw, if you're making use of existing data sources, that may be an efficiency there. So this has to be balanced and kind of thought about ahead of time and what your actual goals are for a particular study. Next slide, please. This diagram is trying to represent the range of real-world data and real-world evidence we may see in clinical studies. It perhaps is more than one dimension here, but we've tried to place this on a single dimension where moving from the left to the right is sort of an increasing reliance on real-world data. So on the left, we have traditional randomized trials, and even those trials make use of real-world data. Particularly, real-world data may be used to select study sites and judge the feasibility of a trial before it ever gets started. And some data for the trial, like medical history, is already taken from existing data sources, the medical records. So even traditional trials make use of real-world data. But moving into the more novel uses towards the middle of this plot, we envision like pragmatic trials where much of the data collection, much of the data for the studies will be from existing sources like claims and electronic healthcare data. Further all the way on the right, we get to observational studies, which obviously represent no intervention on the patient and may make use of entirely of existing data sources or may have supplemental data collection. Next slide, please. Okay, so let me get more into kind of the legalese here and tell you what kind of the world we're living in for FDA, what we're looking for in real-world evidence. So sort of the modern FDA history for drug regulation started in 1962 with this act, and it defined substantial evidence necessary for efficacy. Before this act, drugs weren't actually required to show efficacy in this way. But now this act said you require evidence consisting of adequate and well-controlled investigations. Now, this act doesn't really define it any more than the words that follow in that paragraph. But ever since 1962, there's been further regulations and further guidances to kind of flesh this out. And particularly this adequate and well-controlled investigations are specified in regulations. And that generally, I'll summarize some of the key aspects of that. Generally, those like what we think of as a modern study, like a clear objectives and methods, a well-defined control group. Importantly, it talks about how patients are assigned between the investigated arm and the control group. And it says typically randomization would be used for that. So randomization is mentioned in the regulations, but in the same regulations, it talks about the use of external controls, which sort of perhaps leaves the door open for more observational studies to provide substantial evidence. So this is where the disclaimer comes in mind, because I'm kind of talking about what might happen, not what's actually happening at the moment. I provide this link if you want to see more or learn more about this drug regulation history. Okay, next slide, please.
So Mark, I think I solved
sure.
the, so see if you can advance them yourself, I think.
Okay. Oh, I went the wrong way. Okay, can people see 21st century cures?
Yep.
Okay. Okay, so as I said, 1962, this criteria of substantial evidence was made law, and that's been being fleshed out with regulations and guidances and some laws here and there. And along came in the end of 2016, this large act, 21st Century Cures. This was a large act. This was like the cancer moonshot, if you remember. But among many other things, it asked FDA to establish a program to evaluate the potential use of real-world evidence. And this was mainly to support new indications. So these are drugs that are already marketed but might be seeking a new indication. And also support these post-market studies, which might be for safety and efficacy. So it defines what real-world evidence is here, and it's actually been redefined in the slide I provided earlier. But it talks about sources other than traditional clinical trials. And by other than traditional clinical trials, what's meant here is maybe pragmatic trials or maybe even observational studies. And what's important to note in this act is there's no change in the evidentiary standards. There's no implication or statement that anything is different in terms of the substantial evidence criteria that we've been living with for some time now. Okay, I think I'll advance the slide now. Okay, so FDA has outlined their intended program for real-world evidence in this document that came out at the end of last year. So this is FDA's plan for how to implement the program we've been asked to do under 21st Century Cures. And it's a multifaceted program. There's going to be changes in our internal processes. There's going to be a lot of guidance development, which I'll touch upon on the next slide a bit. There's some stakeholder engagement and some demonstration projects to see what works and what doesn't work. And, you know, if you're interested in kind of the regulatory use of real-world evidence, I definitely recommend you read this document. You can even comment on it for the next week or two. Okay, so just a little bit more on this framework. There's lots of aspects to this framework, but in some ways, maybe it could be summarized in kind of three main components, starting with like whether the data is fit for use. This is like whether you're using a data set for a particular outcome in a study, is that outcome reliable? Does it, you know, validly measure what you're trying to measure? You know, there's we have to know also whether people are taking the drug, and we also need information on covariates if we're running an observational study. So basically, is the data fit for use? And then on the other side is the study design. Is the study design appropriate to provide strong evidence? And if it's for efficacy, again, it has to provide it has to fit under this substantial evidence criteria. So when can pragmatic trials provide substantial evidence, and when possibly can observational studies provide substantial evidence? So that's kind of study design considerations. And on the bottom are regulatory considerations. These perhaps are less statistical, but these are things like when you need informed consent to collect data, when IRBs need to be involved, how the data could be audited, they're important for kind of the nuts and bolts to regulatory processes, but perhaps a little less scientific. Okay, so that's basically the kind of regulatory background, and I'd like to move on to the example, which I hope is illustrative. This is a real example that we have had a lot of internal discussion around FDA of whether a study like this might meet substantial evidence. Whether that's the case or not, it's clearly a very informative study for the particular drug problem we were looking at. So this paper, written by some colleagues at FDA, including some statisticians in my division, looks at some serious outcomes, stroke, bleeding, and mortality for patients on anticoagulants. So this was an observational study, and I'm going to go into more detail. Okay, so a little background. So this is a study of anticoagulants. So anticoagulants are used for a variety of purposes, but the indication we're focused now is for atrial fibrillation patients. So they're indicated to reduce the risk of stroke and embolism in these patients. And these are all approved drugs. They are all approved based on randomized controlled trials, although actually I'm not sure exactly for warfarin, which was approved in 1954. But the ones that are approved in the last decade, these novel drugs, they were all approved based on very traditional trials comparing each of them separately to warfarin. So warfarin, why this is kind of important, these are really very innovative drugs. Warfarin is a very effective drug, but it's a difficult drug to use. Everybody's dose has to be adjusted. You have to be tested regularly to make sure you're on the right dose and your diet affects the dose. Whereas these modern anticoagulants, referred to as NOACs, they have standard dosing either once or twice a day and none of the associated monitoring. So they're potentially very, well, they are very useful drugs, and they're very innovative drugs that have come along in the last decade or so. Okay, this just shows the rise of these drugs. So on the bottom are the rise of the novel drugs, the NOACs, and on the top are the corresponding decrease in this drug warfarin. Can I just check that I'm on slide 15 because I accidentally pressed the page down instead of the icon. Are we on slide 15?
Yep, slide 15, it says study motivation.
Okay, thank you. Because I saw it advanced, I wasn't quite sure. Okay, so the question is, we have these 4 new NOACs. So how do they compare between each other, and how do they compare to warfarin in the real world? So a prescriber and a patient, how do they select among these drugs? The original trials compared them to warfarin, but they were in, as I said very early in this talk, in a very controlled environment, and warfarin is not always used exactly right in the real world. So are there differences between how these behave compared to warfarin in the real world compared to the very controlled trials that were used for their approval? And also, how do you choose among the different NOACs? So obviously this has kind of obvious, important, medical implications. So the data source was Medicare claims. This is U.S. Medicare. Most of the patients who take these drugs are elderly too, so Medicare does do a good job of capturing the patient population that takes these drugs. And we're also able to link Medicare to Social Security deaths, so we're able to get mortality as well. Okay, here I'm trying to describe, well, you have this data, which in one of the first slides, I had that timeline of a patient and all the different data sources. how do you take data like that and make a study? These are become very traditional pharmacoepi studies. So we've been doing these, other parties have been doing these for some time now, for 10, 15 years. They've become very standard. But if you're not familiar with them, so this is basically a study based on claims data. So you take, to even get the drug use, is based on dispensing. So all you know is at some point in time, a patient receives a particular prescription for a certain number of days. And you have to take that data and create some algorithm that will create like an exposure episode. So unlike a clinical trial where you might have like a daily diary of when the patient took it, you have to infer based on what you have when they actually took the data, when they actually took the drug. Of course, this has to be validated. You just can't assume this is right, but we've done a number of these studies, so we understand how this works. So similarly for the outcomes, these are diagnostic outcomes that are associated with some medical event that appears in the claims data. So if somebody is hospitalized and somebody has to, and that appears in the claims data, there's a diagnostic code behind it. And there's other events that might have diagnostic codes behind them. And these codes are used for the outcomes in the study, particularly, the stroke, the various forms of bleeding and death. And again, we can't just assume these are right. There's other separate validation studies to give us confidence that these studies are actually providing valid, this use of the data is actually providing valid outcomes. Okay, I want to leave time for the other speakers, so let me go through this. This is the type of study, again, is this new user cohort that we've been doing for some time. So here, T0 represents the new use of a drug, so whether a NOAC or a warfarin, and there's a look-back period in the claims data to make sure there was no previous prescriptions for the drug, that there's diagnostic codes, that they actually have the disease that we're interested in, and we also use this information to collect covariates to make adjustments, and this is going to be propensity score adjustments. And looking forward, we look for the outcome data or some censoring event. I'm going to skip this, how the propensity score matching, and go right on to the results. Okay, so it's a little complicated table here, and I don't really have a pointer to guide you through this, so let me try to do this in words. So running down the left are the outcomes. So the first box is ischemic stroke, and then we have comparisons between dabigatran and warfarin, rivaroxaban and warfarin, and apixaban and warfarin. And the first column that says Medicare, that's the hazard ratios from the study that I've just been describing. So dabigatran it has a hazard ratio of 0.8, which means the estimate is it has less strokes than warfarin, and there's a corresponding confidence interval. And what the other columns show is what the clinical trials showed when the drug was approved. So that was the randomized traditional clinical trial. So the RELY trial was used to approve dabigatran, and that was a comparison between dabigatran and warfarin. And likewise, each column shows the trial used to approve that drug. And you can see overall, this Medicare study, this observational studies and claims data corresponds well to these trial data results. And if anything, the drugs appear more effective, the new drugs appear more effective in the Medicare study. And we like to think that we hypothesize that reflects the real world use of warfarin, because as I've said, in real world, warfarin is not an easy drug necessarily to use, and these drugs are very straightforward to use. So perhaps you're actually seeing some additional benefit of the drugs in the real world. Okay, I think you have these slides so you can see comparisons for the other outcomes. So let me move on to my quick conclusion here and then yield my time. So my final thoughts are, and these are, I guess, pretty obvious. There's a wealth of RWE. We all know we live in the big data world, and clearly this data is going to be useful to answer important medical questions, and clearly it already has. But how it's going to be used to provide strong evidence, and particularly kind of in the FDA regulatory world of substantial evidence, how that's going to happen is under development, and we'll have to see how that progresses through examples like this and other examples. Okay, so I'll be available at the end for some questions, but I think, Dan, you'd like to move on, right?
Yep, so thank you so much. That was a great presentation, and just what we were looking for in terms of, folks like you who have a lot of experience in this to see how you're thinking about it and how you're actually approaching real examples is great. So with that, we'll move on to David Madigan's presentation. So let me get his slides up here. David Madigan is Professor of Statistics at Columbia University in New York City and Dean Emeritus of Arts and Sciences. Received his bachelor's degree in mathematical sciences and a PhD in statistics, both from Trinity College in Dublin. He's previously worked for AT&T, Soliloquy, University of Washington, Rutgers University, and Skillsoft Incorporated. He has over 200 publications in such areas as Bayesian statistics, text mining, Monte Carlo methods, pharmacovigilance, and probabilistic graphical models. He's an elected fellow of the American Statistical Association, the Institute of Mathematical Statistics, and the American Association for the Advancement of Science. Serves as editor-in-chief of Statistical Science and of Statistical Analysis and Data Mining, which is the ASA's Data Science Journal. So we're very lucky to have him here today. So take it away, David.
Thanks so much, Dan. I actually don't see my slides. Do you see my slides? I'm looking at the last of Mark's slides.
No, I still see my own slides. Dan, are you still there? Yeah.
It's a rather ominous silence.
Okay, I think.
Yes, I'm sorry. I was just on mute.
Okay. I think I switched it on the top. There was this little menu there. It says viewing to Madigan. Can you see that now?
I can, and I'm seeing David's slides.
Okay, I think it's okay now.
I'm not.
Yeah, I'm looking. I don't think.
Yeah, I'm not either. This is Liz.
Okay, let's see if we can fix this. Okay. All right. I think it's okay now. I think it was just a delay in the system.
Okay. Can everyone? Can you see honest evidence, etc. My first slide.
I can see them. Liz, you'll be our litmus test. Can you see them?
Yep, I can.
Excellent. Okay. Thank you. And let's see. Can I control them? So, do you now see my second slide?
We do. So.
Yep. Very good. Thank you. Great. Thank you, Dan, for the invitation. I'm happy to be here and looking forward to some discussion at the end. I'm going to talk about some work that I've been engaged in in the context of the Odyssey collaboration, which some of you may have heard of. And I'll say a little bit more about Odyssey as I go along. So I'm going to be talking about observational studies. I'm going to be talking about exactly the kind of study that Mark just described, so I can go through this quickly. So what's on the screen here is a study that was published a couple of years ago, very similar, actually the exact same context that Mark was talking about. And I've just essentially picked this as a more or less random example of an observational study. So the medical literature is there are many studies like this in the medical literature, and it's these kinds of studies are providing much of the evidence base for the practice of healthcare. This particular paper, it's nicely done, and the main sort of product, if you will, or the output of a study like this, is or are statistical artifacts. So this particular one, for example, I'm showing you one hazard ratio that this paper produced, along with a confidence interval, and it's the confidence interval that I'm kind of primarily focused on here, which is to say. That interval is meant to give you some sense of the uncertainty that remains, or the uncertainty around that estimate of 1.82. And the general idea is, we hope, that 95% of the time intervals like this contain the true hazard ratio. So, a reasonable question to ask in the context of observational studies is, how reliable are they? Which is to say, does that interval contain the truth 95% of the time, or is it something other than 95% of the time? There are also very interesting questions about reproducibility, which I won't focus so much on today, although I might touch on it once or twice. So there are some reasons to be concerned that the reliability might not be what we would like. What I'm showing you here is a picture of the medical literature, so to speak. And I'm going to be showing you a few pictures like this. So on the horizontal axis is actually every dot in this picture is an estimate from a paper in an observational study in the literature, like the one on the previous slide. The horizontal axis here is the point estimate, the estimated effect size, or relative risk, or hazard ratio, or whatever, 1 being the null value. And the vertical axis is the standard error. So you get to be statistically significant if you're under those red dashed lines. And so you need to have a large effect size and or a small standard error to be statistically significant. So this is we just did a crawl of the literature. There's about 30,000 estimates on this picture. 85% of them are statistically significant. So it could be that analysts choose are remarkably selective in what they choose to study and publish. But it's sort of the clustering that happens just below the dashed line, particularly near the center of the plot, near 1, is certainly a little bit alarming. And you say this shouldn't necessarily be 5%, but 85% seems quite extraordinary and potentially troublesome. So why might there be questions about or concerns about reliability? Well, there are many issues to do with that literature we've just been looking at. There are issues to do with multiple modeling and p-hacking and so on, which I'm not going to focus so much on that. But there are also issues with these studies, with observational studies. There are concerns about various sources of error, selection bias, measurement error that Mark actually discussed briefly, model misjustification, multiple modeling, and especially unmeasured confounding. So you're comparing two or more than two, but let's just say two groups of people who took drug A, warfarin, and people who took dabigatran, and you attempt to control for differences between those two groups of people, differences other than the drugs that you're studying. But there's a worry that you're missing things, that there are things that differ between those two groups of patients that you didn't measure or that you didn't adjust for. And that quote from Paul Rosenbaum at the University of Pennsylvania, I think is very apt. So the implicit in these papers is a claim that that there are no unmeasured confounders, and that you're right that there are no unmeasured confounders. And it's a little scary to be relying on human judgment in that context. So what we've been doing in Odyssey for the last couple of years. So there's my mandatory Greek slide. I'm from Columbia, so I have to have a Greek. This is causal inference that we're trying to do here, and it's hard, is what that slide basically says. So what we've been trying to do in the context of Odyssey is we've been studying an alternative approach to what I just showed you. So the current approach is this one-off. You take a question and you design to the best of your abilities. You design a study to answer a single question. What we've been exploring is instead a more systematic approach. So a fully reproducible, systematized approach that's open source, that doesn't study one drug and one outcome, or a pair of drugs and an outcome, but instead studies, essentially all drugs in a given therapeutic area and a large set of outcomes. So instead of doing one study, you do, so to speak, all studies. And that's one piece of our key sort of aspect or facet of our approach. And then a second key facet is the use of negative controls and positive controls. So negative controls, also known as falsification hypothesis, this is not a new idea. Negative controls in this context are drugs and outcomes that are, quote, known to have no causal association. And there's a bit of a circularity here, because how do you know there's no association if we're not certain how to do causal inference? What we're doing right now is we're identifying negative controls using a process that does a crawl of the literature. It extracts information from product labels. It analyzes spontaneous reports. And you can read about it in a couple of papers that we've published. So reasonable people could certainly have concerns about the way we do this, but it's an attempt to identify, essentially outcome, in the given context, outcomes that appear not to be caused by the drugs that you're studying. And with those negative controls, you can generate empirical p-values, which are, you can get an empirical null distribution by basically running your analysis on the negative controls. So you're producing p-values that don't begin with an assumption that you have an unbiased estimator, and that those concerns that I listed on the earlier slide don't exist. Positive controls are trickier, and currently what we're doing is we're generating synthetic positive controls. We take the negative controls and we inject signals on top of them with a known effect size, and that allows us to compute calibrated confidence intervals. So we have p-values and confidence intervals, the primary sort of statistical artifacts, that the idea is that we hope and we can show that they do indeed perform close to their nominal characteristics in terms of coverage and type 1 error rate. So, oops, sorry, pressed the wrong button. A brief word about Odyssey before I show you some of show this paradigm in action. So Odyssey is a collaboration of several hundred individuals all around the world, focused on generating reliable and reproducible evidence from observational data. I welcome you to join Odyssey and take part in their many now working groups focused on all kinds of different topics in this context. The members, people involved with Odyssey, some of them have observational data, or they sit on top of observational data. The union of those databases has a very large number of patients. It's north of 500 million patients. The crucial thing is that these databases have been mapped to a common data model, the so-called OMOP common data model, which enables one to do this kind of research on a very large scale globally. And it's a game changer in terms of what it enables. The Odyssey collaboration is studying a whole suite of use cases for observational data. Population level estimation is what I'm talking about now, what Mark was talking about. There are other activities going on within Odyssey. There's a major activity on patient level prediction, and there's a lot of activity on clinical characterization, using these data to understand healthcare, basically the practice of healthcare. So there's a world beyond population level estimation that I'm not focusing on today. Okay, so how does this work in practice? Well, in the context of a study we did about a year ago, studying a suite of treatments for depression, we developed a set of negative control outcomes. So ingrowing nail, we deemed it pops up as a negative control, so meaning it is an outcome that appears not to be caused by any of the depression treatments that we were looking at. So, how does this p-value calibration work? Well, if you run a crude analysis, I think this is on one of the Truven databases, if you run a crude analysis of this is duloxetine versus sertraline, two particular treatments, for this outcome, what you find is that you get a statistically significant hazard ratio, 1.16. In fact, if you run all of the negative controls that we have for these depression treatments, what you expect is that 5% of the negative controls should have a p-value less than 0.05, in the sense that the null hypothesis is true for all of these. Instead, 68% have p-value less than 0.05. So the type 1 error rate in this very specific context is an alarming 68%. If you calibrate, I should say, they were crude estimates. If you do something more sensible, if you do something a lot like what Mark just described, using propensity-based methods, the type 1 error rate is more like 16%. So, and we found this in general, that crude estimates are really can be very problematic. But using kind of, if you will, a more modern approach, the type 1 error rate in this particular context is 16%. That's still pretty far off 5%, and it sometimes it's worse than that, and sometimes it's better than that in the context we've been studying. If you calibrate, what you do using the empirical null distribution, essentially what happens is the boundary for statistical significance changes. It's now that yellowy area that is on the slide. There's not a huge difference in this particular case between the nominal boundary and the calibrated boundary for statistical significance, but nonetheless, after calibration, the type 1 error rate is 4%. So it's not exactly 5%, but it's a lot closer to nominal than it should be. And this is what we've seen, in one context after another, that the calibration procedures do restore the nominal characteristics. This particular example is not very dramatic. Here's another example in a different context where it's much more dramatic. So it's completely empirical. So those boundaries, from the calibration procedure are not man-made. They're completely empirically driven. Okay. Let me focus from my last few minutes on an application that we haven't published this yet. We've presented it on a couple of occasions. We've been studying treatments for hypertension, and what prompted this is a recent major clinical practice guideline to do with the care of hypertension, published towards the end of 2017. It's a very impressive document. It has 106 distinct recommendations about hypertension treatments and different agents and what have you. The authors were very careful to describe the quality of the evidence that supported the various recommendations. Some derive from randomized controlled trials, but it's a minority of the recommendations. And at the other end of the spectrum, many of the recommendations draw on, quote, expert opinion, which is perhaps a little unsatisfactory. So what we've done, what the Odyssey collaboration recently completed, is a very large-scale study of hypertension treatments. So we're looking at around 40 hypertension or antihypertensive drugs. They're listed on that slide. On the left is the frequency with which these are used in a large claims database in the United States context. So we're studying 40 drugs for hypertension, and so in some of our analysis we group them into classes. We're looking at 58 outcomes of interest. So these are outcomes that are generally the kinds of outcomes that are considered in studies, and they're both good things and bad things that may happen downstream of these antihypertensive drugs. We developed negative controls using this semi-automated procedure that I mentioned previously. So we've identified 76 negative controls. So these are outcomes that we do not believe are caused by any of the drugs that were on the previous slide. So 40 drugs that we're studying, 58 outcomes, 76 negative controls, and then we run this analysis for every one of those. If you do the combinatorics, it's a very large number of analyses. What we run for each one of them is a study. It's a new user cohort design. It's essentially exactly what Mark was just describing. So we have a period before treatment where there has to be no prior antihypertensive drug in that period or no prior cardiovascular outcome. Do propensity score matching, and then we look at on-treatment effects and intent to treat effects. And with the propensity matching, we look at a variety of approaches: stratification and weighting and trimming. So it's very similar to what Mark was just describing. So we perform these analyses in a suite of databases that are listed on this slide. So they're US insurance databases, Japanese, large-scale Japanese insurance database, Korean national insurance database, a couple of electronic health record databases, and a electronic health record database in Germany. So these are databases that have been mapped to the overall common data model. They're in that Odyssey universe that I had a picture of there earlier. So that's the basic idea. On this slide here, you can see what I'm showing on the left is every dot there is one of those 40 treatments. There's a line where, in any case, where for any pair for which there exists evidence from a randomized trial of the comparison of those two drugs. So what we've done basically is fill in that circle. So there are actually 10,278 comparisons that we run for each of those outcomes that I listed in each of those databases. This is non-trivial computing, but it can be done. So here are the results. So at the end, we don't actually produce all of the estimates because in some cases you don't have any power, particularly for the less used drugs and the rarer outcomes. But in total, in this particular study, we're producing about 1.3 million estimates. We're doing 1.3 million studies like the one that Mark showed us. After calibration, about 83–84% of the confidence intervals include 1. Now, that doesn't have to be 95%. That shouldn't be 95% because we believe that some of these drugs do cause some of those outcomes. But nonetheless, that's what the picture looks like when you do all studies in a particular therapeutic area. By contrast with the picture I showed you earlier, let me flick between these two. So that's what the literature looks like, where not just hypertension, but just in general, this is what the literature looks like. So 85% of the results are statistically significant, whereas when you do all studies, no p-hacking, not cherry picking in any way, it's essentially the flip of that, where 15% are statistically significant. So, perhaps this is what the literature should look like. Arguably, that's what it actually looks like. Now, how well does this work? Well, we can look at this, from a variety of perspectives to try and get some handle on how good this is. One thing you can do, we've done this extensively, is to look at how well these analyses perform on the controls, so the negative controls and the positive controls. So what I'm showing you here is from left to right is negative controls are on the leftmost column where the true hazard ratio is 1. Then going from left to right, it's 1.1, 1.5, 2, and 4. So these are the positive controls that we injected. And then the top line is prior to calibration, and the bottom line is after calibration. And what I'm showing you here is a particular comparison, the thiazides versus ACE inhibitors. The MI there is a typo. That shouldn't be there. This is the performance on the control outcomes. So what you see, and so you can look at many classes and many comparisons, but what you see is prior to calibration, the confidence interval coverage varies from 73% for the negative control down to 36% for when the true hazard ratio is 4. So it's far from the nominal 95% coverage. And below the line, you see the coverage following calibration, which is much closer to 95%. And it sometimes it's better than this, sometimes it's not as good as this. It varies depending on the pair of drugs and the database that you look at. But the point is that the confidence interval, the calibrated confidence interval, comes much closer to meaning what it purports to mean than the nominal confidence interval. We've also done some comparisons with randomized trials. I'm not going to go through this in detail in the interest of time, but 28 of the 30 estimates where there is a randomized trial that we can compare with, in 28 of the 30 cases, the estimates are concordant in the sense that Mark described in his context. For 2 of them, they're discordant. So I just make an observation that comparing observational studies with RCTs is a subtle kind of a business. So there are many reasons why an observational study might not agree with a randomized trial. One of them is bias, the residual bias present in an observational study, but there are many other reasons, because as Mark described, observational studies are in the real world, whereas randomized trials are generally done in very selected patients. So it's not crystal clear that this should be the gold standard in evaluating observational studies of this type. Okay, so it's 1.3 million studies in essence that we've done in this particular application. Visualizing these results or navigating these results or searching these results is not such a simple thing. We've developed a couple of tools that I invite you to take a look at. The Legend Basic Viewer. Legend, by the way, stands for Large Scale Evidence Generation in a Network of Databases. The Legend Basic Viewer allows you to drill down on one dot in that picture and see exactly how the analysis was done, and you can look at balance and covariates and overlap of propensity scores and what have you. The Legend Med Central was intended to be a gimmick. It's basically like PubMed Central, so you can actually look at a computer-generated paper that describes every one of those dots that looks a lot like a paper in the literature. So it is an interesting way to think about viewing or visualizing very large-scale evidence on this kind of scale. So that's what I have to say. The international community that's conducting this work, along with this very large-scale global data network, is very interesting as a platform to do research and to understand observational studies and to come up with better ways of doing observational studies. I think it's a very powerful tool that we have at our disposal. I didn't say anything much about the common data model, the OMOP common data model, and there are a lot of people involved in that model. That's critically important, and you can't do this kind of research on this kind of scale unless there's a common data model. And I just finished by saying that I'm not for a second suggesting that we've cracked observational studies or that somehow this is the way to do them. But I think what I do hope is that more work takes place in the coming years on understanding the real operating characteristics of observational studies, and hopefully we keep improving the way we do these studies to generate higher quality evidence. I'll stop there. Thank you.
So that was fantastic, David. Our folks had asked for some ideas of what can be done, and this is just incredible what can be done sort of on a massive scale. So thank you so much for that presentation. Liz, I'm going to pass you the ball, and if you could start sharing your file as soon as I've done it, which is now, because I think part of the issue before was just simply a time lag. So if you could start that process right now, I will move to your introduction, and hopefully we don't have to wait too long. So we're very lucky to have Elizabeth Stewart, who is Associate Dean for Education and Professor of Mental Health, Biostatistics, and Health Policy and Management at the Johns Hopkins Bloomberg School of Public Health. She received her PhD in statistics from Harvard University, is a fellow of the ASA, and received the Myrtos Lefkopoulou Award, the Gertrude Cox Award, and the Health Policy Statistics Mid-Career Award. Her research primarily involves non-experimental methods for estimating causal effects and methods to assess and enhance the external validity of randomized trials. So I think you're in for a treat. What you're going to see in this presentation is a little bit different. It's not so much focused on the treatment comparison that we're so used to, but I think you'll really enjoy it because it really gives you an idea of what sort of work can be done with real-world data out there. And so with that, I think your slides are up, Liz. You're ready to go.
Great, thanks. Thanks for that nice introduction, and I was going to frame it similarly in that where I'm coming from is a little bit of a different perspective, much more public health oriented, and so my goal is to give you very high level, quick summary of a few different sort of applications of these sorts of methods and data that maybe complements, but is slightly different from what you've heard so far, which were both really great summaries of the sort of regulatory treatment effectiveness sorts of contexts. So hopefully when I do this, you will see slide 2. This is just the quick introduction. I will also really try to finish by 12:20 so we can get at least 10 minutes of questions. So I will go through these quickly. The slides will be available after, so you can get all the details then. So this is, I'm going to just quickly go through these 3 different applications of sort of potential uses of real-world data. And again, I'm coming from a slightly different perspective than the previous speakers. But the first is related to what we've just heard, so sort of non-experimental comparison group designs. This might be where there's a single-armed trial going on currently, and then interest in using some historical control data, maybe registry data from the past. It could also be settings or sort of more what David Madigan was talking about, studying effectiveness of treatments in real-world practice, sort of post-approval. And the sort of workhorse tools here are things like propensity score, more broadly comparison group designs, instrumental variables. I'm also then going to talk very briefly about an application of using this sort of data to help understand the generalizability of randomized trial results, both to sort of document similarity of trial participants to populations of interest, and then maybe also to actually project impacts from a trial onto that population, and maybe perhaps by combining experimental and non-experimental evidence. And the methods there are some new methods, and again, I'll have sites for all of these for generalizing or what's called transporting effect estimates. And then finally, I do a lot of work in sort of policy evaluation or health services research. And these sorts of data sources are used frequently to study, say, different payment models for mental health care or a policy change in some hospital system. And the workhorse tool here are a broad class of interrupted time series methods. So again, I'm going to just try to give you a flavor for each of these and some references to get all the nitty-gritty details. And again, I don't think I really need to cover this because actually Mark did a really nice job, so I'm going to skip that. So the first approach I'm going to talk about is this general idea of non-experimental comparison group designs. And David alluded to a lot of this and had nice examples of both the challenges, but then potential some solutions to helping deal with this. But the fundamental idea is one where you just sort of have a group of people who got some program or policy or treatment of interest, and then a group of people who didn't. And you use what I'm trying to call equating methods. I sometimes used to call them matching methods, but really it's not always matching. Sometimes it's weighting. So I'm trying to use the term equating methods, where the idea is to make exposed and unexposed, or exposed with one drug compared to exposed with another drug, look as similar to one another as possible. There's a huge literature in this area, and again, we've sort of already heard a little bit about this. Of course, the main problem in these non-experimental contexts is confounding, that the people who get one treatment or program often differ from those who don't. As you already heard, propensity scores are a commonly used key design tool for these studies. And the way you can think about it is that the goal is to sort of make the groups look as if it could have been a randomized experiment, at least with respect to the observed covariates. And we don't have time to get into all the details of this, but of course it's really important to note that this relies on the assumption that there's no unobserved differences between the groups once we condition or equate the groups on the observed ones. There are sensitivity analyses that you can do to assess robustness to this assumption, and I'll come back to this very briefly later. So I'm just going to really quickly step through an example where we were using real-world data from Denmark to look at the effects of a suicide prevention program that Denmark had started. So basically, in 1992, Denmark started rolling out suicide prevention clinics around the country. Those are now nationwide. And there was interest in comparing the outcomes of people who received or who went to one of these clinics after a suicide attempt and compare them to people who didn't go to one of those clinics. I don't have time, but the Danish registry data is very intensive, extensive data on basically the entire country of Denmark. So in terms of real world data, it's pretty real world, at least for the country of Denmark. So again, the problem is, this is a very high level summary of propensity score methods. The problem is that we would sort of like to be able to find people who did and didn't go to these suicide prevention clinics who are similar to one another on a large set of observed characteristics. However, it's really hard to do that when you have, in our case, 31 covariates. So you can think of propensity scores as a particular type of dimension reduction where you summarize all of those covariates by one number, the propensity score, which is the probability of receiving the treatment, in this case, the probability of going to one of these centers. And then you can match or weight or subclassify using just the propensity score rather than having to deal with each covariate individually. And there's a couple of nice properties of the propensity score and sort of a nice theoretical underlining to it, which makes it work well in practice. So again, in this case, we had data from 6,000 people who had gone to one of these suicide prevention centers. And then we had about 60,000 people who had attempted suicide but had not gone to one of the centers. And this registry data we had data on individuals, their family structure, their medical history, really extensive data. In particular, what was really great about this data was it included information on their previous suicide attempts, the method of the attempt, family history, including whether their parents had attempted suicide, and psychiatric history. So I don't have time to get into all the details, but we used propensity score matching, and so what we did was for each person who went to one of the clinics, we found 3 individuals with similar propensity scores who also matched exactly on these 2 variables that we felt were particularly important to get good balance on. And actually, I'm going to skip this and go right to this figure, which shows it's a nice sort of diagnostic that can be done, and David alluded to balance checking. So what this is showing is for each of the covariates, each row, this shows basically a sort of effect size. So the difference in means divided by the standard deviation of each covariate. The blue circles are before the matching, so you can see that there are some pretty big differences. There's a sort of rule of thumb that you want this to be less than about 10%, so that's that dashed vertical line. And the red circles are after matching, and we see that the matching has been successful in making these groups look more similar. So the idea is that this propensity score matching has substantially reduced the confounding due to these 31 characteristics. We then conducted survival analyses using these matches, and I don't have time to get into the details, but some evidence that this program reduced risk of repeat suicide attempts, death by suicide, and death from any cause. And again, I don't have time to get into the details, but you can do sensitivity analyses, and so we were able to show that these findings were quite robust to a potential unobserved confounder. We had a really good set of confounders observed, but you're still, there's always that nagging concern, in this case, maybe that there's some unobserved genetic factor or something like that. And we were able to show that the results would sort of stand up even if there was such an unobserved confounder. So again, so the main idea here is that this policy was being rolled out across the country, and we were able to use, quote, real world data, this registry data, in order to look at the effects of this program. In general, propensity score methods are useful for improving estimation of causal effects. And it fits into, there's a growing field of what's called trial emulation, which really goes back to the ideas that Paul Rosenbaum and Don Rubin have articulated for years, which is that in non-experimental studies, it's just as important or potentially more important than in a trial to lay out very clearly the exposure of interest, outcome measure, the timing, covariates, et cetera. And we were able to use this approach in a very large-scale population-based data set. And so we're able to draw inferences about the effects of this program in the population of people in Denmark. But again, of course, we might still be concerned about unobserved confounders. Okay, I know this is a whirlwind, but I'm going to shift gears now and talk a little bit about generalizing trial results. So as I believe Mark mentioned, one of the potential interests in real-world data is that we are, there's growing awareness that the people that participate in trials are often non-representative, and they, sort of they are conducted in certain places, and they take people who agree to be randomized. And there's growing questions about how well those results would translate to target populations of interest. So there's a growing body of work trying to use real-world data to help understand that. The fundamental idea is to either weight the trial sample to look like the population, or perhaps use model the outcome in the trial and then use that sort of outcome model to predict outcomes in the population. A different strategy, which I'm not going to talk about today, but I wanted to briefly mention, is that in this context, we're thinking about a case where we don't have evidence, or we don't have sort of information on effects in the population. Maybe it's a new therapy that hasn't been implemented yet. There's a different strategy called cross-design synthesis, which can be used when you actually have both experimental and non-experimental evidence. So I think that's a really great, interesting field. It's not what I'm talking about today. So really quickly, this is just a quick example of doing this, where there was a randomized trial of, this was an old randomized trial, therapy has changed dramatically recently, but a trial that examined heart therapy for HIV compared to standard combination therapy with a very large effect, a hazard ratio of 0.51. But the question that we asked in this paper was, what would the effects of heart be if it were implemented nationwide among people who were newly diagnosed with HIV? So we basically, you can sort of stack the data where we have the trial data, and then we have a data set representing the US population of newly infected individuals. And you can see in the data, I don't show you here, but the trial and the population did differ substantially on variables that also moderate treatment effects, in particular age, race, and sex. And so for that reason, we might think that the trial result wouldn't directly necessarily carry over to the population. So the approach that's often used is to, again, as I mentioned earlier, weight the trial subjects up to the population. So very similar to non-response weights for surveys or propensity score weighting. We can basically weight the people in the trial by 1 over their probability of being in the trial, given the covariates. There are some variations on this now. I'm giving you sort of the simple version, but the sites at the end have somewhat more sophisticated versions. And again, the idea is to use those weights when comparing outcomes in the trial, and that allows an estimate of the effects in the population, adjusting for those covariate differences between the trial and the population. Again, there are also outcome prediction model approaches that people are exploring, BART and TMLE and others, but I'm not going to focus on them today. In this case, there's a key assumption, which is that it's similar to unconfoundedness, but here the assumption is that there's no unobserved effect moderators that differ between the sample and the population. If that assumption holds, then we can interpret the results as the population average effect. So this just shows the sort of result you can get if you compare just the first row to the last row. The crude trial result was a hazard of 0.51. If we adjust that to the age, sex, race distribution in the United States, we actually ironically end up with a hazard ratio very similar. Things sort of cancel out to give us actually a similar result in the end, a wider confidence interval. But this can be interpreted as a sort of predicted average effect in the population of newly infected people across the US. So again, it's possible to use real-world data to help understand how results from trials would translate or sort of carry over to a population. I do want to clarify the third bullet, that these methods are relevant when you're interested in a population average treatment effect. So maybe for cost-benefit sorts of reasons, we want to sort of project what would be the overall average benefit. These methods are not designed for sort of personalized decision making. This is more about estimating a population average effect. Okay, 5 minutes. I'm going to just quickly, this one is very short, just give a really quick flavor. A completely different use of real world data, but honestly is one that I probably use the most, is for cases where we're interested in studying studying an abrupt policy change. And there's a whole, again, broad class of methods called comparative interrupted time series to estimate the effects of that policy change. This might be a state that changes some law. It could be a health system that puts in place a new policy and wants to study the effects of that policy in their population. It's very common to use large-scale administrative data for these. Two quick examples are the effects of, I worked on a study where we looked at the effects of a federal mental health parity law on mental health service use, and I'll show you a picture from that in a second. And then also we've done this looking at gun control laws. And both of these we're using sort of, large-scale real-world data. The gun control, for example, was using counts from the FBI and sort of publicly available data from national sources. So just to say a little bit more on the mental health parity study, in this case, we were using market scan claims data, so healthcare claims data from a very large sample of people across the US to look at the effects of this federal law on kids who have autism spectrum disorder. Our sample size was about 40,000 kids, and we were using an interrupted time series model, basically looking at trends over time in service utilization. I don't have time to get into it here, but there's lots of complications in using this sort of data, including things like with claims data, you have people coming in and out of the sample based on when they have health insurance under this system, and so you have to worry about sort of selection that might be happening based on who's in the sample. Ideally, you have the ability to define covariates based on some previous period, and so you need longitudinal data for each person. Of course, there's confounding. The claims data itself is unstructured, as we heard a little bit about earlier, and so for this purpose, you need to decide, well, should I model it weekly or monthly or quarterly or annually? There's sort of lots of questions of how to get from the initial data you start with to the analysis sample. In this case, we used monthly counts, and we were using interrupted time series ARIMA model to model trends over time and basically project out what would have happened in the absence of the intervention. This was not an ideal study because there was no comparison group. The federal policy impacted everyone in the US, so it wasn't as strong but it was sort of the best we could do. So this is the sort of result you can have. So the yellow line is the observed data. This is the number of outpatient visits that these kids were getting. And you can see the yellow line is what actually happened. The red line is sort of what we project would have happened had parity not passed. And so the difference between the yellow and red lines on the right-hand side is the estimated effect of the policy. And so this is the sort of analysis that can be done to look at these abrupt policy shifts. Okay, in 1 minute. So again, I just want to come back to some of the challenges. I told you some of the opportunities. Now some of the challenges in using this data. I alluded to this already, but measurement timing is messy. One particular complication when studying some policies or programs is that there's no kind of index date for comparison group members. So we see the date that a treatment group person, gets the surgery or starts taking a medication. We don't get to see that date for people who maybe could have started that therapy but didn't. That's why sometimes a cleaner design is to compare drug A to drug B. But in some contexts, you really want to be able to compare drug A to kind of not taking any drug, but then you need sort of a way to define a kind of baseline index date for the comparison group. And there have been advances in that, but that's something that comes up in practice. Measurement error, we've already heard a little bit about, and sort of the measurement error, I liked Mark's reference to fit for purpose. The measurement error is going to have different implications depending on whether the variable is a covariate, the exposure, or an outcome. There's also a lot of challenges if you're trying to combine data, like the trial and population combining, because often things are not measured the same in those different sources. And so there's some harmonization that has to happen, and that is not always easy. Of course, there's missing confounders and moderators. And just an aside, I will say, as someone who sits in public health, I actually think that epidemiologists have thought a lot more about some of these kind of these sorts of issues then have statisticians. And so the epidemiology literature is actually one that has a lot of papers sort of just talking about some of these nitty-gritty challenges. So again, lots of opportunities and plenty of challenges too. There's a huge literature on these topics. And I, just to this group, I would say let's make sure that statisticians are at the table when thinking about how to use or not to use the data. There's a little bit here, and then I have a couple of slides with references, and again, these will be available after so you can see all of them. Thank you very much.
Great, great. Thank you so much. What a an excellent presentation, and again, with a little different view of things, especially with the health policy side. We do have a few minutes for questions. Many of you who have been part of NISS meetings before know that we usually had 2 speakers and a discussant, and today we decided to go with 3 speakers because I really thought that everyone would enjoy hearing all 3 topics. But if you have any quick questions, please submit them now. And before we get there, since we do have a little extra time, I guess I have a question I'd like to ask each of the panelists in turn to answer, which is, part of the part of the genesis of this particular meetup was that folks wanted to hear from people who were sort of in the trenches dealing with real world data. And I was wondering if each of you could comment on, since you all have that experience, what are the 1 or 2 things that, having had that experience, you'd like to sort of relate to the rest of the group, or those of us who don't have much experience in that area, of things that you found that either were surprising or particularly challenging or anything like that. So Mark, why don't we start with you?
Okay, well, I'll just, one thing that comes to mind is maybe what Liz alluded to at the end of her talk is the involvement of epidemiologists in this field. And that's generally a good thing. I think she was mainly referring to the methods development. But this sort of work requires a real understanding of the data sources and what they represent, like the clinical context of where these data come from. So I think before you get too far into these studies, you have to ensure that you have the kind of necessary expertise to understand the data sources and the clinical context. And that's typically epidemiologists or clinicians that would provide that. I think probably, as you see from David's talk, there's a lot of computational aspects as well. So that might be another unique aspect to these studies.
Great, thank you. How about you, David?
I guess the one thing that comes to mind is the complexity and subtlety of defining outcomes. Is something that I certainly didn't realize before I started working in this area. So you want to even sort of fairly hard outcomes like MI. Trying to capture that as best as possible, in observational data is not a simple thing. And just looking for an isolated ICD-9 code or ICD-10 code, whatever, is might be fit for purpose, but is often not fit for purpose. So it's a tricky business. I just throw in one other thing which I failed to mention in my talk, which is we don't often pre-register observational studies, and I think that is critically important. We're going to have to start doing that to be credible, for these studies to be credible. Even today, the pre-registration is not the norm. It is for clinical trials, as we all know, but not for observational studies.
Great, thank you. How about you, Liz?
I'll ditto what was just said by both Mark and David. I totally agree with both of those. And actually, just to expand one thing on David's point, one thing I've always been interested in is that I think for a non-experimental study, in some cases, it may make sense to pre-specify a process for picking an approach rather than a specific approach. And I just say that because, at least in my experience, I never can predict in advance what propensity score method is going to work best in a given data set, matching or weighting or whatever. And a nice thing about propensity score methods is you can check and you can pick them based on, say, covariate balance. So I just want to make a plug for thinking about pre-specification of a sort of decision rule, where maybe it's going to be, I'm going to try these 3 approaches, and I'm going to pick the one that, wins based on some covariate balance metric. I think it's just a little bit of a different kind of sense of pre-specification, but I totally agree with the general premise. The thing I'll just add is to expand on the comment I made earlier about how important things like the common data model are that David alluded to. I've been incredibly frustrated it's been really difficult in cases where we've been trying to generalize randomized trial results to target populations. Often the limiting barrier is data and sort of not having consistent measurement of the covariates between data sources. It's incredibly difficult to get them to be similar. So I think the more trialists can work to kind of have measures that are common and then can really help enable lots of use of that data and with other sources, I think the better it will all be. So just a plug for those efforts at sort of data harmonization and standardization.
Yeah, and I'm thinking of the common data method that David alluded to and how difficult that must have been. And I'm sure we could have a whole series of meetups just on that topic, and it would be not hard to fill it all. Okay, so I see we're just about to the end of the allotted time. So the first thing I want to do is really give a hearty thank you to our presenters. They were 3 excellent presentations. And just to remind folks that, the presenters do this simply as a service to the statistical community. And so when you see them next time at a conference, buy them a cup of coffee or do something, because really this is the type of collegiality that we like to have, obviously, as statisticians, but it's the type of information that's hard to get elsewhere. So thank you to the presenters, because it really is doing a great service to the community. Last but not least, I want to mention that, thanks also to Christy Trang-Stein for helping organizing, and Kara for being my technical assistant here today. And then just to remind you that we do plan to come back with a meetup probably in the 3rd quarter of this year. It's taking shape, and it's likely to be on subgroup analysis. So lastly, let me thank the attendees. It certainly is great to have folks interested in these topics, and we look forward to next time. So with that, I'm going to sign off, and I hope everyone has a great day. Take care.